about our next unit 1.3 membrane structure. So let's see the understandings. Phospholipids form bilayers in water due to amphiphilic properties of phospholipid molecules. Membrane proteins are diverse in terms of structure, position in the membrane, and function. Cholesterol is component of animal cell membranes. And now application and skills application. The cholesterol in mammalian membrane reduces membrane fluidity and permeability to some solutes. And skill here involved is drawing of the fluid mosaic model and analysis of evidence from the electron microscopy that leads to the proposal of the Debs and Danieli model, and analysis of falsification of the Debs and Danieli model that led to the Singer-Nicolson model. Guidance. Amphiphilic phospholipids have hydrophilic and hydrophobic properties. Drawing of the fluid mosaic model of the membrane structure can be two-dimensional rather than three-dimensional. Individual phospholipid molecules should be shown using the symbol of the circle with two parallel lines attached to a range of membrane proteins be shown, including glycoproteins. So membrane structure. As early as 1915, scientists were aware that the structure of the membrane isolated from a cell included proteins and lipids. Further research established that the lipids were phospholipids, and early theories were mostly concerned with the phospholipids forming a bilayer with the proteins forming thin layers on the exterior and interior bilayers. The Davison-Danieli model proposed by Hugh Davison and James Danieli in 1935 used this lipid bilayer model, uh, suggesting it was covered on the both sides by a thin layer of the globular protein. In 1972, Seymour, J. Singer, and Garfield Nicholson proposed that proteins are inserted into the phospholipid layer and do not form a layer of the phospholipid bilayer surfaces. They believed that the proteins formed a mosaic floating in a fluid layer of the phospholipids. There were several reasons why Singer and Nicholson proposed a model that was different from the Davison-Danieli model. These reasons included the following things. Not all membranes are identical and symmetrical, as the first model implied. Membranes with different function also have a different composition and different structure, as can be seen with the electron microscope. Protein layer is not likely because it's largely non-polar and would not interface with water as shown by the cell studies. Much of the evidence used to change the Davison-Danieli model was gathered with the use of the electron microscope. Another source of evidence was study of cells and their action in the various environments and solutions. The ability to culture cells in the laboratory allowed many of these studies. Since 1972, further evidence has been gathered about the membranes and slight changes to the Singer-Nicolson model have been made. The current agreed model for the cellular membrane is the fluid mosaic model. It is shown in the following diagram as the cellular membrane, whether plasma membrane or the organelle membranes have the same general structure. Glycoproteins are composed of the, as we can see in this figure, are composed of the carbohydrate chains attached to the peripheral proteins. They play a role in the recognition of like cells and are involved in immune responses. And the integral protein shown in the red color is completely penetrate the lipid bilayer. They control the entry and removal of the specific molecules from the cell. And the phospholipid bilayer, as you can see here, and the cholesterol, which is shown like in yellow color, helps to regulate membrane fluidity and is important for membrane stability also. And some polar substances, particularly ions and carbohydrates, are transported across the membrane via the channel proteins. Some non-polar substances are transported directly through the lipid bilayer. And phospholipids in figure 1.14 note that the backbone of the membrane is bilayer produced from the huge number of molecules called phospholipids. Each phospholipid is composed of a three-carbon compound called glycerol. Two of the glycerol carbons have fatty acids, and the third carbon is attached to the highly polar organic alcohol that includes a bond to the phosphate group. In this figure, we have seen the fluid mosaic model of the cell membrane. There is double layer of lipids, the fats arranged and their tails facing inwards, like their tail was facing inwards, and the proteins are thought to float in the lipid bilayer. Okay, so fatty acids are not water soluble because they are non-polar. In contrast, because the organic alcohol with phosphate is highly polar, it is water soluble. This structure means the membrane have two distinct areas when it comes to polarity and water solubility. One area is water soluble and the polar and is referred to as hydrophilic, water loving. Hydrophilic is water loving and hydrophobic is water fearing. This is phosphorylated alcohol side. The other area is not water soluble and is non-polar. It's referred to as hydrophobic, that is water fearing. And here we have the model of phospholipid, organic alcohol attached to the phosphate group. The glycerol fatty acid are connected like this, and the polar hydrophilic region is this, and non-polar region hydrophobic is this thing. So in the diagram, as you can see, the hydrophilic phosphorylated alcohol heads the shown here, and hydrophobic region of fatty acids here, and hydrophilic phosphorylated alcohol heads we have here, just like these are same. And the model of phospholipid bilayer shows how phospholipid molecules behave in the two layers. Both layers have the phosphorylated alcohol end of the molecules towards the outside, and the fatty acid tails oriented towards each other in the middle. So cholesterol must be fluid to the function properly. They are a bit like olive oil in their consistency at various locations in the hydrophobic region. Fatty acid tails in animal cells are cholesterol molecules. These molecules have a role in determining membrane fluidity, which changes with temperature. The cholesterol molecules allow membrane to function effectively at a wider range of temperature than if they were not present. Plant cells do not have cholesterol molecules. They depend on the saturated or unsaturated acids to maintain proper membrane fluidity. The proteins, the last major component, it's the major component of the cellular membranes comprising the proteins. It is these proteins that create the extreme diversity in the membrane function. The proteins of various types are embedded in the fluid matrix of the phospholipid bilayer. This creates the mosaic effect referred to the fluid mosaic model. There are usually two major types of the proteins. One type is referred to as the integral proteins, and the other type is referred to as the peripheral proteins. The integral proteins show an amphipathic character with the both hydrophobic and hydrophilic regions with the same protein. These proteins will have the hydrophobic region in the midsection of the phospholipid backbone. Their hydrophilic region will be exposed to the water solution on the either side of the membrane. The peripheral proteins, on the other hand, do not protrude into the middle hydrophobic region but remain bound to the surface of the membrane. Often, these peripheral proteins are anchored to the integral protein. Look at the drawing of the fluid mosaic model 1.14 to see the location. You can look at the figure 1.14 to see the location of the proteins. So membrane protein function, as you will recall, it is the membrane proteins that impart different functions to the different membranes. There are many different proteins which have the six channel function, sites of hormone binding and uh, enzymatic action, cell adhesion, cell to cell communication, channel for passive transport, pumps for active transport. Proteins that serve as hormone binding sites have specific shapes exposed to the extrovert and that fit the shape of the specific hormone. The attachment between the protein and the hormones causes the change in the shape of the protein, which results in the message being relayed to the interior of the cell. Cells have enzymes attached to the membranes that catalyzes many chemical reactions. The enzymes may be on the interior or the exterior of the cell. Often these are grouped so that the sequence of metabolic reactions called the metabolic pathway can occur. Cell addition is provided by proteins that so that they can hook together in various ways to provide permanent temporary connections. These connections referred to as the junctions can include gap junctions, tight junctions. Many of the cell-to-cell communication proteins have carbohydrate molecules attached, and they provide an identification label that represents the cell of different types of species. Some proteins contain channels that span the membrane and providing passageways for substances to be transported through. When this transport is passive, material moves through the channel from the area of high concentration to the area of lower concentration. In active transport, the proteins shuttle a substance that one side of the membrane to the another side of shape. The process requires the expenditure of the energy in the form of ATP. It does not require a difference in concentration to occur. And so we have some interesting exercises, questions that you can try. So our next unit will be membrane transport that we'll be looking in our next video. Okay, so thanks, and that's all for today.